pressure you put on me, and I'll do as you please. Great Spirit, blow me in the breeze. Spirit over me like bamboo in the wind. I've been, 'cause I'm planted so deep, I can feel the touch. I'm deeply rooted, I can feel it, oh, I can feel it all. I can feel it, oh, I can feel it in my bones. I'm deeply rooted, I can feel it, oh, I can feel it all. I can feel it, oh, I can feel it in my bones. Washing over me, so deep, in my bones, washing over me. Great Spirit, I feel it in the wind. So deep, I can feel the touch of Spirit over me like bamboo in the wind. I've been, 'cause I'm planted so deep, I can feel the touch. I'm deeply rooted, I can feel it, oh, I can feel it all. I can feel it, oh, I can feel it in my bones. I'm deeply rooted, I can feel it, oh, I can feel it all. I can feel it, oh, I can feel it in my bones. So deep in my bones, washing over me. So deep in my bones, washing over me. I know pressing into the soil. I'm flying out of my mind and into my heart.
And welcome to OMSI's Virtual Science Pub. My name is Rebecca, and I am the Teen and Adult Engagement Assistant Manager at OMSI. And for tonight, I am your host. Before we get started, I have a fun few things to tell you about what's going on at OMSI these days. First of all, OMSI is theoretically open. OMSI is open not during inclement weather. Our planned reopening happened right during the snowstorm. But as soon as our building has power again and the ice has receded, we will be open. The Genghis Khan exhibit just closed, but our Natural Sciences Hall, as well as the planetarium, and submarine will both be open. Our next exhibit, Dinosaurs Revealed, will open on March 20th. Advance tickets are highly recommended, and please check our website for the most up-to-date information about if we're actually open, when we're open, and how to buy tickets, and our COVID safety guidelines, all at omsi.edu. I also hope you enjoy tonight's pre-pub trivia and music by local Portland artist Trinetti. We hope to have them at a Kendall concert in 2021 sometime. Putting on these live shows takes a lot of work, and we have an amazing partner that helps us make this happen. So a big thanks and shout out to Cellastream for providing live streaming services for tonight's Science Pub. We really appreciate their support. Oops, wrong one. And also, if you want to do fun things with OMSI from home, we have some really fun stuff for you. Our 6th virtual OMSI After Dark is the Winter Ale Fest, and we have curated a really awesome box of 10 different winter beers, including 4 stouts, 3 porters, and 3 other winter ales. So please join me. I'll be one of your co-hosts that night for some wonderful science demos. We'll interview the brewers. We'll have some really cool entertainment, some magic, some music, some jokes, some laughs, and it'll be a wonderful virtual event that'll happen Friday, February 26th. Tickets are $54 and are available at www.omsi.edu/alefest. Also, if you can't wait until February 26th to have some delicious ales, you can put the pub back in the Science Pub experience by ordering some delicious food and tasty beverages from these food and beverage partners we have around the state. Hopefully they have power and are open right now. Maybe wait till they get power back and support local businesses. For tonight, what we'll be doing is our typical Science Pub program, more or less. And what that looks like is we will start with a neuroscience themed trivia game. That'll be a warm up for tonight's talk. So get your trivia brain on and get ready for that. And after trivia, we'll have a lecture with Dr. Lockwood. And then after the lecture, we'll have question and answer time. And if you have any questions at all throughout any point in the lecture, feel free to submit those questions via the comments in our live feed. And we will collect all those questions. And then after the lecture, I'll ask our speaker. If you enjoy tonight's lecture, please consider making a donation or purchasing a Science Pub pint glass, which I have here. And I tell you, it's really cute, it's really great, and it's like very hefty. It's good quality glass. So I recommend Science Pub pint glass, $5 suggested donation. You can also donate without, if you have enough pint glasses around, you can donate anyway. But there's no pressure to donate. Our mission at OMSI is to inspire curiosity, and we'll do that, regardless. So please sit back, relax, and get ready for a great lecture right after our trivia. This week for trivia, I would love to welcome on Alex Mastinduno, our chemistry lab coordinator at OMSI, and who's gonna win all the things with all of the stuff. Hey, Alex, how you doing?
Hello, I'm good. How are you?
I'm doing great. I'm glad you're here and that you have enough power to connect to your computer.
My mother-in-law has enough power for me to connect to my computer, but yes.
We're all gonna get through it.
No.
Does your brain feel ready for some brain-themed trivia questions? How do you feel about brains?
I feel about brain. I am interested, but I wouldn't say that I am overly knowledgeable already. So
Ooh, okay.
I will be increasing my knowledge through this game. It's gonna be wonderful.
This is great. So for folks at home, I hope you're ready to join us for trivia. If you want, you can shout out your answers like Alex is going to, or you can say your answers sedately like Alex will probably do. You can shout them out if you're a person who doesn't have a sleeping baby. You can also type them in the chat if you want to participate. Then we'll see what people's answers are in the chat. Super fun. How this will work is we'll have 10 multiple choice questions. I'll read out the question, give you time to guess, and then I'll reveal the answer to each question before we move on to the next one. Does it sound good? Do you feel ready? You pumped? Okay. Question
Let's go.
number 1. Which sensory system or systems, so this is a choose all that apply situation, are not contralateral, which means controlled by the opposite side of the brain, where like the right thing is controlled by the left side of the brain kind of thing. So which systems are not like that? A, smell, B, taste, C, vision, D, hearing, or E, touch. I see like
I know I'm
crossing
about senses and like organs which belong to which things. Yes, you've got 2 eyes, 2 ears. Many people have 2 eyes, 2 ears, 2 sensory touch receptors. I'm gonna guess. I'm also gonna a little sort of meta wondering about the topic of tonight's theme. I'm wondering if it's both smell and taste. So A and B.
Is that because we've got one tongue and tonight we're specifically talking about smell?
Yes.
You are so good. Look at those powers of deduction. I love it. Yes. Smell and taste are not controlled by the opposite brain half.
That's so interesting. I didn't know the other senses work. That's cool.
Yeah. Crazy. Okay. How many different odorants, smells, fancy science word for smells, sniffers, can the human nose distinguish? I guess how many smells can your sniffer detect? Oh, this is terrible. I need to start putting more words onto this. A,
nope,
500,
this is great.
B, 10,000, C, 5 million, or D, 1 trillion.
Well, first of all, I'm only calling a nose a sniffer from now on. And I'm thinking it has to be one of the higher 2.
There's a lot of smells out there in that world.
There's a lot of smells. I'm go big. I'm gonna say a trillion. D.
A trillion is correct.
Yeah? A trillion? Really? Actually?
1 trillion. I know.
Wow.
I've heard different scientists have different opinions, and I don't know. That's a fact I knew that a few years old. Maybe there's more of a consensus now. But they're like, I think once you get up to that many numbers, can you name all the smells? I don't know. Maybe we'll find out.
I could name a trillion anything
in the lecture. I can name $1 trillion. What is another name for an ear, nose, throat doctor or scientist? An otolaryngologist, a nasologist, a septumologist, or Dr. Oz? And don't make your judgment on how much trouble I had saying the word, because that's not indicative.
We're all sounding out. That's great. I'm going to just go ahead and say not Dr. Oz.
Mhm.
I assume. I feel like he's a surgeon.
He's on the TV for something.
Yeah. I think he's got an actual doctorate of something, though. I am going to say the otolaryngologist. That sounds like a word that I've heard before.
And it's the one that has more than one thing mentioned, like ear, nose, throat.
Oh, good point. Good point.
I'm just joining you in guessing, which is not coaching.
That's great.
The person doing the trivia, but
That's
it's
okay.
true.
It's team trivia, Rebecca. We can be a team.
We are a pretty good trivia team. As
That's
long as
true.
the subject is Harry Potter.
It's Harry Potter. That's what this is about, right?
If anybody at home wants to bet money on Harry Potter trivia, Alex and I will win your money. So don't worry about that.
As long as Jen is with us, yeah.
Who invented the Kleenex? Is it A, Kimberly Clark, D, Dr. Cleanest, C, the U.S. Army Corps of Engineers, or D, those folks in the USSR?
I love the name Dr. Cleanest. I like that it wasn't just Dr. Kleenex. Wonderful use of the product. I am going to say, I think it'd be funny, the U.S. Army Corps of Engineers. I think that would be entertaining for some reason.
That would be entertaining.
Is it? No, I thought that for a moment, but perhaps not. I really, what I meant to say was A, Kimberly Clark.
Yeah, you would be right. I don't know who Kimberly Clark is, but hopefully we'll learn more about this in the lecture, because I believe we have another Kleenex themed question next.
Okay.
Which, yeah, Kleenexes and noses. Actually, you can see I am using a Kleenex on my nose. Actually, I think it's Puffs. Technically, I'm using a Puffs. Anyways, which comic book character was featured in Kleenex ads? Is it Popeye, Little Lulu, Captain Future, or Mickey Mouse? I believe this is an old-timey comics question.
I was going to say, based on the look of that ad there, I'm going to, yeah. Let's go Mickey Mouse.
That is the most recognizable comic book character.
Okay, I was really thinking more something along the lines of, that's also recognizable, Little Lulu.
Yeah. I don't know who Little Lulu is. Apparently this
I don't know, but she's got amazing little ringlets. Look at that.
I'm into this whole fashion situation she's got going on. So yeah, it was Little Lulu. Okay, moving on. We're halfway through. You made it through halfway, and I believe that you have 1,000% correct. So
boom, nailed it. I'm doing so good. I'm feeling really good about myself.
when was the first computed tomography, or CT scan of the
That's what CT
brain
stands for.
performed? Was it in the very far long ago, 1936? Was it in the much less far long ago, 1971? Even newer, 1985? Or fairly recently, 1992?
36 seems like, sorry?
I was going to say, I don't know if this photo helps you at all.
It is less helpful to me, I think, than the last one, but 36 seems like too far long ago. I'm going to say 71.
I hope I didn't lead you astray, but I thought this picture looked kind of 70s-ish. Yeah, it was. 1970.
Boom.
We've had CT scans for a long time.
That's cool. I didn't know what CT stood for. Computed tomography. Cool. I learned.
Yeah, I feel like, and maybe Dr. Lockwood will correct me later, but I think when it was CAT scan, it was like computer assisted tomography. Maybe not. I might be making that up. I should stop. Okay. Number 7. Where was the first
Where?
CT scan of the brain performed? Which scientists in the 70s did this thing? In A, England, B, Russia, which back then was the USSR, C,
Mhm.
the United States, or D, China?
For no particular reason, England seems like the right answer.
Yeah, they did a lot of crazy stuff there. I think that's where x-rays started too, and the heart. What is the thing that does your heart with the boop boop? Anyways, yes, you're right, England. Yeah, there you go.
When you started talking, it made me think that I needed to choose a different one again.
Oh no, sorry, England was right. I was just very excited about some other fun facts.
That's awesome.
Yes, England. Okay.
Yay!
What travels fastest? The Bugatti Veyron, which I'm not a cool person, but I think that's a car. A neural signal. C, a diving peregrine falcon, which is a bird. B, the SSC Tuatara, which I have no idea what that is, but I feel like it's mechanical and not biological.
I'd buy that. Mm-hmm.
It's a car. It's a sports car. My sources tell me.
The Tuatara is a sports car?
Yeah. Uh-huh.
I'm assuming the Bugatti as well. I'm going to say a neural signal. I feel like those are fast and topical.
That would be cool. I don't know if I can nudge you correctly for this. I'm just going to tell you. It's
okay. With the Falcon was going to be my next guess.
Yeah. It's this car. It's a really fast car, which is surprising. I feel like neural signals are electricity.
That's what I assumed.
This is another thing I want to hear more about. I've already got so many questions about this lecture. I'm really excited for it. Okay. Fun tree picture. Okay. The Greek picture for the branches of a tree. Wait, I don't know why I said picture. The Greek word for branches of a tree gives us the name of what part of a neuron? Soma, axon, dendrite, or synapse?
I feel like I actually know this one because a dendrite is like the this shaped thing. Mm-hmm. I'm going to say dendrite.
Yeah. And dendro is the tree word. Dendrite. I learned that from Can't Hardly Wait, a fine film from the late 90s. Okay. Number 10. Last question. You're doing so good. I hope everyone at home feels as great as Alex feels right now. Which lobe of the brain is primarily responsible for our ability to smell? And there's a helpful diagram there if you're not sure where things are. Is it A, the parietal lobe, B, the temporal lobe, C, the frontal lobe, or D, the occipital lobe?
Oh man, I'm nervous to say where my thoughts are going because I don't know if they're right. For some reason, I have it in my head that the occipital lobe has something to do with sight, so maybe it has something to do with other senses. I'm going to say occipital lobe.
That is the one that has to do with your sight. The smell, it's not necessarily.
It's not necessarily the same thing. Parietal.
I can't remember, so let's see. It's temporal.
Ah, darn. Okay, nope.
I feel like that's also where memory. It's like smell, taste, memory. These things.
Okay, because sense memory.
We'll learn more about this also because I believe Dr. Lockwood also studies memory and senses. So this is really exciting. Okay, Alex, you are amazing. Thank you so much for playing trivia with me.
It's my pleasure.
I hope you have a wonderful rest of your night and kiss that baby.
I shall. Pretty great. Okay. Bye. Thank you so much for having me. I'll see y'all on a future one of these probably.
Yeah. Okay. Okay, folks, it is time for our speaker, and I would like to introduce Dr. Denesa Lockwood. She is, oops, I just lost my notes. Hold on, everybody. Dr. Denesa Lockwood is a behavioral neuroscientist who studies behavior and the underlying neural mechanisms by which behavior arises. She graduated from Western Oregon University and then completed her graduate work at Florida State University with a dissertation focused on conditioned taste aversion. She currently studies the genetic underpinnings of alcohol addiction, but remains interested in learning and memory, including sensory memories. When she's not getting mice drunk in the lab, whoops, hold on, I lost my thing again. Denesa stays busy wrangling 3 children, 2 dogs, and a constantly varying number of fish. She has been an active volunteer and a voice for broader science dissemination in the community, heading up the March for Science efforts here in Portland for several years, and she is also a board member for the nonprofit Educational Outreach Northwest Noggin, where she has volunteered across the country teaching students of all ages about the basics of neuroscience. She's also recently in 2019 received the Sigma, I already forgot how to say this, Sigma XI Outstanding Supporter of Research Award. I'm not good at reading Greek. Sorry, everyone. Okay, that's enough talking from me. Hey, Denesa, how you doing? You feel ready? Oh, I think you're still muted.
Hi.
Yeah.
Hello. Thank you. Thank you for everyone
Welcome.
for coming.
Okay, Denesa, take it away.
All right. Well, thank you. That was so fun for the trivia. I have some corrections, which are embarrassing to admit since I wrote the trivia, but I will discuss those as I go on here. And so I'm going to share my screen and see if I can figure out how to start my presentation. Let's see. Does that look okay?
You are shared. Yeah, that looks great.
Okay, awesome. So the title of my talk this evening is Can You Smell Me Now?, which I thought was sort of a clever allusion to the old Verizon commercials, until I realized in talking to my children that maybe no one remembers those commercials, where of course the tagline was, Can you hear me now? Anyway, I wanted to talk tonight about how COVID-19 affects both our senses of smell and taste. And I did want to just disclaim here that although I do work for OHSU and I study behavioral neuroscience there, I'm not a licensed medical professional, and all of the opinions, of course, are my own. So with that being said, I think we can carry on here. These are some pictures, of course, of lots of things that I mostly stole from the internet, but some of which that I've generated. And one that is me here. And I get to wear a mask every day, which is nice. And also this very fancy helmet, which helps keep me safe from allergies that I have to mice that I work with. So I am very used to wearing the mask that, of course, we're all used to. And this is another little statue on campus, which is now covered with a mask. So getting started, I want to just give a brief overview of sort of the problem that we're addressing, which is kind of this notion of a lack of smell or anosmia. And one of the things that I love about this field is sort of where these words come from, and they often come from Greek or Latin. And so A, of course, is without, and then nosmic comes from this notion of smell. So here we have without smell. And even as early as March of last year, this was recognized by the president of the Rhinological Society in England as one of the main distinctive symptoms of COVID-19 infection. And it's often present without any other symptoms. So that was sort of curious in people. And there's a wide range of numbers. And so we won't talk too much about statistics in this talk, but depending on who's counting or how recently the paper has been published, these numbers are changing all the time, but it seems like a high number of people are reporting this anosmia or a lack of smell. And so it can be as high as like 60%, but there's a wide range, like 15 to maybe 86% of people who report this. And then additionally, 67%, again, maybe there's a range there, of patients report diminished taste. And so that's agousia, which we'll talk about a little bit later. So we know then that hyposmia, which is a diminished taste or a lowering, smell, a lowering of smell, was also reported during the first SARS infections that occurred in 2002. and then other types of coronavirus infections, which can result in things like the common cold. But the curious thing about this COVID-19 infection and anosmia is that it does seem to come on very, very early before other symptoms arise, and then also, like I said, without any other symptoms. So we're kind of curious why that is. Before we talk about smell too much, though, I wanted to talk a little bit just about definitions and taxonomy, because I am always curious about how things are named, and I think part of the problem is sort of labeling of the problem. And one of the things that we know is that the genetics of each of these viruses can help us in treatment. So this is a schematic of all of the different coronaviruses that were identified at the time of this drawing. This drawing is actually before our coronavirus had emerged. And so it is in this beta class, or this beta genus, actually, of viruses. And one of the things I wanted to point out was that if you look at this tree, there are a couple of others here that are of note, which is this Middle Eastern respiratory syndrome coronavirus. And then this one here, this human coronavirus 229E, which causes sort of a mild cold-like symptom. But we see that there's tons of these which are named for the animal which they infect. So there's a peafowl and a partridge, and lots of bats, and bats. So I was sort of wondering, and again, I am not really an expert, I'm not a virologist, but as I was studying this, why there are so many of these coronaviruses which infect bats specifically. And one of the main ideas is that bats really live in sort of dire conditions where they have these huge colonies and they live very densely populated, and they sort of travel through wide territories. And so they're always being exposed to new pathogens, and then they bring those home immediately to their whole colony, which is sort of like humans, at least in this age of globalization, where we travel wide and far nowadays, and then we come home to our sort of densely packed urban environments. However, bats have sort of evolved for a very long time with these pathogens. And so for the most part, bats really don't become infected in the same way that we do. They're not really dying. There's not a lot of lethality. So they have a very different immune system that we do. And of course, one of the problems is that as these viruses cross from the bat or maybe another species to human, we are very newly exposed to these pathogens, and we don't have that same type of evolution over time to deal with these pathogens. So in looking at the genetics, we can sort of see like who's been afflicted and maybe what the symptoms are. But it also helps us to come to a better understanding of how it works and maybe what the treatment can look like. And of course, then this is very important for solving this pandemic that we're in now. And so just a few other like etymology things. Corona, of course, refers to the crown of the spike proteins that everyone I'm sure is very familiar with at this point. And then for the most part, I thought it was interesting that virus taxonomy or virus naming is not really well regulated. There is an international committee on viruses that's been around for like 60 years, but they don't really have a consensus. Apparently, they were supposed to vote on a new system of naming viruses in 2020, but then, of course, this bout of the coronavirus came about, and so everyone sort of became preoccupied, and that vote had been postponed. So generally, they're named for the species that they infect, and then also maybe the symptoms that they produce in humans, because of course we are the most important species. So in this instance, sort of this new variant of coronavirus is based on the previous one, which had jumped from bats, like I said, in 2002. So we have this severe acute respiratory syndrome, but since this is the second iteration of it, now we're going to call it number 2. So actually the virus goes by the name of SARS-CoV-2, and then the disease that it causes in humans is COVID-19. And I just wanted to sort of take some time to draw attention to that, because I think those terms are used sort of interchangeably, and they're not really the same thing. So the virus itself is SARS-CoV-2, and then the disease is COVID-19. And probably for this talk, I will say coronavirus, even though that, applies to this whole family, because SARS-CoV-2 seems sort of unwieldy to me. But all of these are RNA viruses, which are very different than from DNA viruses, which include herpes and some variants of hepatitis. And then there are some viruses who have both RNA and DNA, and those include like HIV and hepatitis B. So those are going to be in different families altogether. And of course, we're not going to talk about those tonight. So now that we sort of know why we're calling it what we're calling it and why it's called that, how does this work to create COVID in the body? And the main understanding that we have now is that this viral particle, the SARS-CoV-2 or coronavirus particle, has these spike proteins that can bind to one in our body called the ACE2 receptor. And so this is sort of a complicated system that I won't get into too much tonight, but the gist of it is that we have this system in our body that is basically for regulating blood pressure. It is implicated in all sorts of different organs, not only just the nose or the mouth, which we're going to talk about tonight. There's some in the brain, there's some in the heart, lungs, colon, and other systems as well, kidney. And this is why COVID-19 is such a detrimental and damaging disease, because it does have these little molecules, this ACE2 receptor, all over the body. So when the virus enters our body, it is able to kind of bind this ACE2 receptor in all of these different places and sort of wreak havoc. And we think of it as sort of a key and a lock then. But the system is kind of interesting, and it has a lot of feedback. And the reason I'm sort of going through this is because then this also informs where we might be able to develop therapeutics or ways to stop this from damaging the cells in our body. So this ACE2 protein stands for angiotensin converting enzyme. So it's an enzyme that does a job. Usually enzymes kind of cut something up. And this one is 2. So there's another one, angiotensin converting enzyme 1. There's no 1, it's just without a number. But it converts angiotensin into angiotensin 2. In my opinion, this is all very confusing because everything is sort of the same name. But I thought this diagram was helpful. So this one, the first enzyme, we kind of think of him as maybe, or she, the bad guy. It converts angiotensin into angiotensin 2. And then this angiotensin 2 goes on throughout the body in all of these different places to create sort of things like this. So you have like blood vessel constriction, oxidation, inflammation, apoptosis, that's cell death. This could be very bad, and you could see where this could lead to all the symptoms we see, including lack of smell. These things can be exacerbated by some genetics or maybe lifestyle choices. But luckily, there's kind of this natural braking system or a feedback mechanism. So we have this ACE2 enzyme, angiotensin converting enzyme 2, which instead turns these proteins into another form of angiotensin, or actually 7, or I think actually there's like 9. And those go throughout the body and kind of do the opposite. So they create, blood vessel dilation. You have oxidation kind of turned off. Cell death is turned off. And so this is kind of a good guy, right? So this ACE2 receptor, this enzyme, we want that to work. And we need this to work as well, because you can imagine sometimes you need to turn this on, like in fight or flight situations. And so this feedback is very important. The system is very important in the body. And this virus, with maybe evolution or just by luck, has figured out a way to sort of hijack this one particular protein in our body, which happens to be located everywhere, And what happens is when that binds to that receptor can't do its job. So if this is not doing its job, and these things are not happening, then of course you have more of this side happening. And so you have increases in blood pressure, you have increases in cell death. And this is probably the mechanism where a lot of symptoms, not just loss of smell, body.
Hey folks, I think we're having a few technical difficulties on Denesa's side. So hold tight while we try to unfreeze her. While we wait to get back up and running, I believe we'll play a little bit more pre-show trivia. Is that correct? Am I saying the right words here? I think so. Cool. Okay, so hang tight and we'll try to get Denesa back with us soon. Hey, this is what's fun about doing things from home, right? Okay. Apologies there. As you might have heard during the intro, we have had some pretty bad weather in this part of the world, and that I think has affected all of our internet and power. So I'm going to keep going. And I think where we left off was maybe here with the dog nose. So one of the things I wanted to correct quickly was that I did write those trivia questions tonight, and I know that I put the answer down as 1 trillion chemicals or odorants that humans can detect. Actually, this is pretty widely contested, as our host mentioned. Many people who study olfaction for a living, which again is not me, don't think it's that many. And there are lots of reasons for that. And the study has lots of sort of flaws that maybe we can talk about later. But this number seems to be quite a bit lower than 1 trillion. So the general estimate that I think most people are holding is 10,000 to 100,000 chemicals. However, that's still quite a lot, I think. And like Rebecca noted, we probably can't even name all of those. So I think we have a pretty good sense of smell, and we're very sensitive. We can detect things that are really low threshold or concentration, and we can determine things that are very, very chemically similar apart from each other. So I did kind of want to talk about other animals a little bit at the end, because I do study animals, and I think they have really important and interesting senses
that we can learn from. So we are not really macrosmatic anymore. We're kind of microsmatic. We do have a keen sense of smell. It could be considered crucial to survive, but really for us, I mean, it's more subjective and it's more emotional, and it has a lot to do with hedonism and sort of how we enjoy life. And I think that is really important, and we'll sort of talk about that with this loss of smell and why people have such a hard time with it. But obviously, you don't really need smell to get yourself to Trader Joe's and buy your frozen orange chicken or whatever. So just as kind of a primer, a little overview of our human olfaction system. So here we have a diagram where the base has been cut in half, basically, and we see this nasal cavity here, so things are coming in the nose. That's air and odorant molecules, or these chemical molecules that are going to determine what we smell and when and how and where. And in that space, that nasal cavity, right up at the edge is this kind of layer of mucus membrane, some people would call it, but it's also called an epithelium. And that has these very nice little shiny, I don't know if they're shiny, but they're very specific and cool and sensitive cilia. And those cilia are attached to these olfactory sensory neurons, or olfactory receptor neurons. And those go right up through the skull into this olfactory bulb. So one of the things that's different about the olfaction system compared to other sensory systems is that it's really just like a one-shot transmission of information. It's very rapid and it's very close to the surface. So we'll talk about taste and how it differs a little bit at the end, but with smell, things come in, they get processed, and they go right up to the brain. It's very fast, and that's good, and it also can be bad, as we'll find out, because it's sort of like this very easy way for foreign things to enter, and to maybe even enter into our brain, which can be problematic, but might not come into play so much here with loss of smell, but can be problematic for other pathogens. So that is the overview for that. And then as we move along, when I said I wanted to talk about other animals, one of the pictures I had here was, this is from a rat. And you can see that the olfactory bulb, which I just showed you on the last slide, is up sort of inside the skull, is much, much bigger than our olfactory bulb in a human brain. Oh, the label didn't show up, but here's the bulb here on either side. And you can see they're very small compared to our very large and lovely brain, which we sort of think is superior, but our sense of smell is definitely reduced in proportion compared to that of other animals. So at any rate, this is the bulb. So those olfactory sensory neurons have traveled now from your nasal cavity up into your skull, inside the skull there, and they're going to synapse with the olfactory bulb. And then that's going to project deeper into the brain to lots of other brain regions. So remember in trivia, we talked about the temporal lobe. This is the underside of the brain. The temporal lobe is where then a lot of processing occurs. So you sort of have this direct transmission happening right away. First synapse comes in here, and then the brain's going to do some fancy processing, and that's going to enable us to perceive. So this is the sensory maybe component, and then this perception is going to happen deeper in the brain, where you really think, oh, that smells like roses, and I remember I had roses one time for prom, and I hated that guy I went to prom to, and that's going to, unleash a whole series of memories. So just speaking to that, then here is sort of a diagram of all the different brain regions that are involved in that olfactory processing that occurs. There's sort of a sensory component with these olfactory receptors, and then the bulb, and then we're going to go deeper into the brain, these other areas of the brain, which may be implicated as well in this lack of smell. So if any of these things don't work or become dysfunctional in some way, you can see how there's a lot of pieces, and then that would actually render you sort of anosmic, or maybe partially anosmic. And so we have some MRI data just sort of showing you the proof there that different areas of the brain sort of light up or become activated in response to odorants. So if you present an odorant to a human when they're in the MRI, these are the eyes here, and this is the front of the brain. You can see these areas of the brain sort of become highly activated. So this is called the orbital frontal cortex, sort of where we make our decisions. Some higher level processing, kind of the executive part of the brain, sometimes it's called. And then here in the temporal lobe are these areas called the piriform cortex and the amygdala. And so as those are red, you can see that those are really highly activated in response to odorants. So zooming back out a little bit, I wanted to talk a little bit more about what's happening down right in the very beginning in the nose, in the nasal cavity, because it's not just those olfactory sensory neurons doing all of the work. We always sort of talk about neurons like they're the star of the show, especially as neuroscientists. But there, of course, are lots and lots of other supporting cells that are there, that are present. And one of the things that we found, and when I say we, I guess I mean, science in general, the bigger royal we. I did not do any of this work. So this was a group from University of Iowa, and they put out a paper in 2017. So this has been some time ago now, but this is after the first SARS infection occurred. Like I said in the beginning, there was some lack of smell noticed in those patients as well. And there are sort of these specialized bundles that happen here in the olfactory bulb, which are kind of special. They're called glomeruli. And glomeruli, again, is sort of a weird word. And basically that comes from the Latin for ball of thread. And I always like that because you can see they do really have this very nice circular arrangement if you look at a cross section under a microscope. So all of these olfactory sensory neurons are coming up here through the brain, through the skull, into the olfactory bulb, and they synapse with additional cells in these glomeruli. And this picture doesn't show it, but this sure does. There are lots and lots of cells around the glomeruli. And so those are very specialized cells, but they're not the olfactory sensory neurons. They're like supportive cells that do other things, and they provide sort of support for those sensory neurons. And so just like any support team or infrastructure, you need all of those pieces to work as well. And so during that paper, those authors were able to infect mice with the first variant of the coronavirus, which occurred back in the early 2000s. And so after 11 days, they were able to look inside this nasal epithelial layer in those mice. And what they found was that the olfactory sensory neurons were intact and the glomeruli were intact, but there's sort of this disarray or dysfunction that's happening in all of those surrounding cells. So that's sort of interesting because we sort of think, well, if the neurons get damaged, then the whole show is lost. But that wasn't really the case. The neurons really seemed to be okay, and it was these supporting cells that were damaged. And so that's going to become important as we sort of revisit this with our SARS-CoV-2 infection that's occurring now in patients. In addition to all of these supporting cells, there's a lot of other specificity that happens once you sort of look deeper and deeper in that what we've been talking so far about has been this olfactory epithelium. So this is a layer, like I showed you in those previous diagrams, that's very near to this part of the skull and the olfactory bulb and the brain. But your nasal cavity is quite large. Inside your face, behind your nose, there's a big hole, and it's coated in what's called a respiratory epithelium. So this is another barrier layer, or mucous membrane, and it contains all sorts of other cells, which are not sensory neurons per se, but they perform very important tasks. And so these guys here are sort of the basic first level of detection. They do this odor detection. And then as they go up into the olfactory bulb, in those glomeruli, those circles there, we see some of these other types of cells. That has to do with like our odor processing and our coding. But down here, throughout the rest of your epithelium, you have some other cells which actually don't go to the olfactory nerve at all, but are innervated by another nerve called the trigeminal nerve. So I'll talk about that a little bit more when I talk about taste, but there are 12 cranial nerves that sort of innervate our face and convey a lot of sensory information. The olfactory nerve is number 1, and the optic nerve that runs our eyeballs is number 2. And then as we go down the line, kind of closer, I guess as you go down vertically, the trigeminal nerve, I think it's 5. I should probably know that. But it is one that mediates both taste and smell. And it actually kind of conveys not really odorant detection, but this perception of like feeling. So if something is warm or cold or tingly, that's conveyed by this trigeminal. These cells here convey that information. So is loss of smell with this new bout of COVID-19 infections due, like we saw before, to the supporting cells or the olfactory sensory neurons, or perhaps to these other supportive cells that are in the respiratory epithelium? So a group recently set out to study that. Oh, I just wanted to summarize a little bit sort of what I said about some of the clinical aspects of this. We talked about how it was reported first, can be the only symptom. This is sort of curious too, because in other viral infections, the loss of smell generally arises because you have maybe an increase in mucus or this increase in inflammation or even cell death. But when doctors are examining COVID patients, they're not really seeing those other things happen. So we know that anosmia can be caused by all these other things, but it often looks much different. Again, it's coupled with other symptoms, or maybe it takes a really long time to repair. This is again sort of a range of numbers related here, but a lot of COVID-related anosmia often resolves in a very short time. I know some people will have long-lasting loss, and we will talk about that. But generally, there's like maybe no other symptoms. And then sometimes people have this loss of smell and maybe some other symptoms, but there's an inverse relationship. So in looking a little deeper at that, I'm not going to go through all of the figures, but I wanted to summarize this recent paper because it uses a new technology that I get sort of excited about because I perform it in my lab. And this is called RNA sequencing. And so this is a summary of a lot of data, and I won't talk about all of it, but what happens is instead of looking at a cross-section of cells under the microscope, like I showed you a couple slides ago from the mice with the first coronavirus type of infection, this actually enables us to look at the inside of the cells, right? So we're looking at the RNA or the DNA, the genetic material that occurs inside of the cells. And this is a very refined way of looking at sort of what the cell is planning on doing. So in those other slides, I showed you how there are so many different types of cells. We have our olfactory sensory neurons. We have all these supporting cells that occur. These are all happening in the olfactory epithelium and that respiratory epithelium. So using RNA sequencing, we can take a biopsy of cells, which of course is a big soup. It's a big chunk. It has all of those things, and they're all combined. And then we can take out just the RNA, and then we know from looking at the genome which of those are which based on specific markers. So we know olfactory sensory neurons look one way. They have a different gene makeup, different code than maybe these basal cells, for instance. So we can separate those cells into different populations based on their genetic code. And then we can look at only one population. So is the RNA or the genetic material in this population the same as that, or vice versa? So this type of technology offers a really, really refined way of looking at what's going on in a very, very small population of cells. The gist of this is that our old friend, ACE2, that receptor that I talked about with the angiotensin, this converting enzyme, is almost not present at all. In fact, the authors in several instances say zero in these olfactory sensory neurons. So again, just like in the previous paper, but using a different technology, we see sort of the same thing. This entry point that we know the virus binds to is almost entirely not present in these olfactory sensory neurons. But looking down here at sort of the summary, so this ACE2 is represented in red, and this is the different kinds of epithelial layers. So we have the olfactory and the respiratory. Remember, that was the two populations sort of inside your face of those mucous membranes. And so if we look at just the red bars, this ACE2 is quite high in all the different cell types in the respiratory epithelial layer. But in the olfactory epithelial layer, there's hardly any in this olfactory sensory neuron population. In fact, almost none. So who cares? What is the point of this? The point is that, again, these main detectors of odorants, the ones that we think do most of the transmission of information, are not likely to be affected by the coronavirus because they don't have the key, the lock. They don't have the lock for the viral key to sort of bind to and open up. So how are we getting this anosmia? How are we getting this lack of smell? Most researchers now conclude that it must be because of these non-neuronal cell types, which I think it's very important because, again, for years and years, neurons were the main things that we studied. And of course, we didn't have this technology years ago. But obviously, there's a huge population of other players or other cells. And so we need to start learning, how do we maybe treat these cells? How do we look at these cells? You know, if something like this is what's causing anosmia, can we deliver a drug, for instance, just to this cell and not that one? Can we target this in a specific way so that, you know, this isn't affected and this is? So that's why these kinds of studies are important, and that's why I think they're exciting to share with you all. And this is sort of a pretty picture from that same paper where they use fluorescent tags, and those are able to bind to these proteins that we've been talking about. So this is that same angiotensin converting enzyme 2. And we see that here in green. It sort of surrounds those glomeruli, and it's really high in sort of that supporting cell layer. And then if we look at these basal cells here in red, we can see that they sort of overlap. And so that's kind of a proof. Not only can you get it if you smush up everything and just look at the RNA, but you can also go back and look at the functional protein in the cell under the microscope. And we see that, yes, this ACE2 really doesn't seem to exist in the olfactory sensory neurons. So they're doing a good job. They're providing sensory information, but they're not the only ones there, and they're not the only ones affected by these type of viral infections. So now that we sort of know the mechanism, what do we do about it? How do you treat this? If you have anosmia, what does it look like? And people who have had a lack of smell know that it basically sucks. In general, you sort of don't like all the things you used to like, you know, like food, especially beer, things that we're probably enjoying tonight seem less enjoyable because that smell component is so huge in determining what we like and don't like. It also can have more severe implications for people. So it's not just the subjective quality, and that's what I wanted to talk about earlier when I said that we don't really need smell to live, but we kind of do need smell to live, you know, to like really enjoy life. It can also create problems for people in, you know, maintaining their personal hygiene. This can lead to things like depression and isolation if you can't share some of these subjective experiences with your friends or family. It can also be really dangerous, of course, if you can't smell like a gas leak or something spoiled. You may die or you may become really sick. So this is a big clinical problem, and it's not only from viral infections, of course. People who have all of those other causes could suffer from anosmia. So what do we do about it? Luckily, with COVID, like I said, most people have a full recovery within a matter of weeks. Some have some long-lasting effects. Those are pretty minor. And generally, one of the cool things about the olfactory system is that those neurons do regenerate, and the supporting cells do regenerate. So with enough time, even if it's not the short duration, you will probably regain your sense of smell. But now there's something else happening, which I had never really heard of before, but I'd heard about it with taste, but not smell, that some things are sort of cross-wired. So something that used to smell good, for instance, chocolate, now is going to smell disgusting, like there's garbage. And this is called parosmia. And this is usually temporary, luckily, but this is really, really severe in people. And they really find it's almost worse than having no smell at all, this kind of mix-up, because some people even suffer where tap water, for instance, smells bad, and then they can't shower without having really, really aversive experiences. So we think, remember how I talked about all of the different olfactory sensory neurons? They have these little receptors on them. And we have about 400 different receptors that can convey information. And each odorant, or smell, is sort of coded in a different way. So for instance, this odorant, whatever it is detected by receptor number 3, slightly. And then this one, this odorant, is detected by not only receptor number 3, these are sort of arbitrary numbers, but also 25, and very intensely. So you can imagine that if you had some damage to those cells that are helping to support the olfactory sensory neurons, you might get your wires crossed, literally. And this could disrupt this type of coding, where now, for instance, this molecule might smell like this one, or vice versa. Or you can see any number of iterations where this type of coding, which is pretty complicated and sophisticated, can become altered in the brain and sort of result in one smell smelling like the other, which can be sort of detrimental. So the treatments then that are suggested, I think are sort of, they're not great, but luckily, like I said, for most people, this does seem to be temporary, at least in the terms of COVID and viral infections. So a lot of it has to do with the volatile nature of odorants. So that means, these chemicals, they are released into the air, which is how we're able to breathe them in through our nose. So if you can prevent that from happening, if you don't heat things, for instance, they're less likely to volatilize. And so they might smell less strong. This is helpful for some people. So you can avoid fried foods, meat, eggs, coffee, chocolate, pretty much all the good things to eat, which sounds horrible, of course. You might try to just eat a diet like this, with plainer things that don't smell as strongly. Some people have to resort to like protein shakes. And then there's this thing called smell training, which I thought was really interesting, especially as we talked about at the beginning. I really do like to talk about memory and how you can relearn things. So you can sort of fix the wires in your brain or in your nose by doing some training. And so there are other things that can help in other causes of anosmia, for instance, steroid treatment, surgical treatment. These are not really as applicable for COVID. But this olfactory training, it's very widely used in the UK. So I put some websites here where there's quite a lot of description. I don't know why it's more widely used in the UK than here. Maybe somebody else in the audience could address that for me. But we know that smell training works because we can see from experiments like this that there are actual changes in the brain that happen. So in this experiment, this is from 2015. So again, not really related to this round of like COVID-19 per se, but in other types of anosmic patients, you can take a batch of healthy controls and then anosmic patients and give them some type of examination. This is ear, nose, and throat. And then you can ask them what they can smell, and then you can give them an MRI. And then we go through this series of olfactory training trials. So for about 12 weeks in this instance, subjects were asked to smell various chemicals, I think twice a day for a very short duration. And then this examination is repeated. And what these researchers found in this instance was that, let's see if I can remember this label now, because I don't have my notes in front of me, that our anosmic controls before training sort of look like this with some activation. But then after training, down here on the bottom, you can see that there are much greater points of activity in this MRI image. This is kind of summarized data. And it shows you that all of these different parts of the brain become sort of activated over time. And so we have not only just our basic olfactory network, which is those kind of olfactory sensory neurons, but then this somatosensory network, which has to do with that trigeminal information. So more of those warm and cold feelings, those also start to increase after training. And then this is an integrative network where things sort of come together and you put all of that information together. And we know that those are different parts of the brain sometimes than just the basic sensory processing. So those also increase, which is encouraging. So we know that olfactory training can work in terms of brain activation. It's actually pretty easy to do. You can buy a set of these sniffing sticks, but a clever name, for a relatively low price. I found them online for $325. They are interesting because they offer you not only a level of different scents or odors, but also different concentrations, right? So we know that you can smell something at one concentration, it might be pleasant, but of course, if it's too high of a concentration, sometimes that's unpleasant. And so that's important to distinguish, that it's not only just the odorant itself, but maybe how much of the odorant is there. So these types of sets are available. It's sort of like a marker with a cap, and each odorant then can be presented to the person for, I think in this instance, they recommend just a real short little duration, just 20 seconds each, 2 times a day, for at least 3 months though. It does take a little while for this kind of rewiring to occur. However, there does seem to be very few adverse effects. It is combined sometimes with cognitive therapy. So not only do you present the odorant, but you are sort of encouraged to imagine that scent. So for instance, if number 1 was lemon or something, you would imagine lemonade and summer and sort of all of that contextual information that you could to help those deeper parts of your brain then sort of send feedback to your sensory neurons at that outset or the beginning of the process and help them sort of rewire. So that does seem to be pretty helpful. And so if you don't have the patience to wait, or you do have long-lasting anosmia, then this does seem to be a pretty effective way to alleviate it. And it does help for parosmia as well. So moving into taste. The taste part of this is a little less complete, mostly because when people clinically report a lack of taste, they usually almost always mean lack of smell. Because what we think of as taste is, of course, a combination of smell and taste. So it's really a lack of flavor that's happening in patients. And that's because this nasal space here, that includes the mouth and this epithelium that we talked about earlier, it gets information from all of these different places. So what we think of as taste, I'm kind of putting that in air quotes, it really does have information from this olfactory system as well. So it's really important when patients say that they have a lack of taste to maybe do some more specific testing to rule out other things that might be going on. So although in recent kind of, clinical journals, there is a high number of patients who report this ageusia, which I think is such a great word. So this is without taste, ageusia, or dysgeusia, which is altered taste. Often oftentimes this is almost entirely related to smell. And the way that we can rule that out is if a patient is able to go in and do some kind of, for lack of a better word, like blind taste testing with very simple taste hints, like that would be bitter and salt and sweet. They can generally taste those, but they feel like they can't because they're lacking this olfactory information. So if patients really do have a lack of taste, that might be something else going on. However, we will talk about how it could be related to this lack of smell or anosmia as well. But as an overview, the taste system, it does seem to be a little bit more resilient to injury, which is another reason that a lot of times patients aren't really experiencing a lack of taste. There are several different nerves that convey this information up into the brain. And those are those cranial nerves that I talked about earlier. So there's the facial nerve, which is 7, trigeminal, I'm pretty sure is 5, this glossopharyngeal nerve is 9, and the vagus is 10. All of these then take information and are innervated by the tongue and the palate and other areas of the mouth and take taste and information up into the brainstem and into the brain. As we talked about before, the trigeminal has to do with like spicy or hot, tingling, burning, cooling sensations. And also, the mouth in general is just kind of a more multifunctional place. And so it does seem to have some redundancy there, which is helpful because not only do we use it for feeding, also it's important for digestion. You have the salivary glands there and other things happening, but speech. So I think in most patients, again, I'm not a medical doctor, we're not really seeing a lack of taste per se or damage to tastent receptors. So let's talk about those and let's talk about how you would determine that. Just like with the sniffer sticks, you can sort of do the same thing with taste, and you can assess that with various solutions that are very specific. So they only contain, for instance, one chemical that would be like citric acid or sour, or sucrose would convey a sweet taste. And so what is happening then is the patient is given like a drop of each solution and they're asked to report, is it salty, sour, bitter, sweet, or no flavor? And then how strong is that? And so this will actually tell you objectively if you're able to determine those taste hints. And then often what's happening is this doesn't really match your subjective reporting. So as you go about your day-to-day, you think you can't taste, but really if you went into a lab and had this tested, you probably could taste. This also can be coupled with some genotyping, which helps to really narrow things down and get specific in that we know some genes that are very, very specific for certain chemicals. And so one type of person might have the alleles. Remember those big A, little a alleles from genetics that allow you to taste, for instance, this chemical called PTC. Some people can taste this, some can't. And so this type of genotyping, if we can see if you are a taster or a non-taster for PTC, enables us to really say with certainty if your taste receptors are working or not. And one of the problems, of course, with COVID is that most of us are experiencing all of these symptoms and told to stay home because the virus is so contagious. So a lot of times you're not really able to do this specific type of like psychophysical testing in the lab. But I just wanted to point it out in showing that there is a specific way to sort of rule out some of these deficits that we think we're experiencing. And then we can sort of treat that patient better depending on what's actually going on. So just like with lack of smell, dysgeusia or ageusia has many different types of causes, not just COVID or coronavirus infection. So I've listed quite a few of these here. These then can be treated. Oh, let's not talk about that, I guess. Let's talk about all of the different types of things that are happening in your mouth. So I mentioned the cranial nerves. And in addition to that, there's tons of different spaces and specialized cells and things happening in your mouth. And they all sort of have different cell types. So we talked with the sense of smell. about those olfactory sensory neurons and then the supporting cells. Well, in the mouth, we have taste and kind of sensory neurons as well, but each type of tissue has its own type of sensory, or I'm sorry, not sensory, but supporting cells. So if we're looking at sort of how lack of taste occurs, if it really is lack of taste and not lack of smell, can we see if the same player is involved, right? Because we know that the virus particle binds to this ACE2 receptor. Is that even present in the mouth? This is not as widely studied. And so that's another reason why it's not as good of a story yet as the one with smell, because we just don't really know. We don't have as much information about some of these proteins and where they are in the body. But there is some work being done. So recently, this paper just came out last year, and a lot of the papers, which I'll list at the end, of course, are very recent because this is all new in terms of this type of infection. But what we have here are just the ACE2, again, shown in green. Green and red are very popular fluorescent tags for this type of work. So this is going to bind in areas of the tongue in this instance. And then these just show the nuclei. And so we can see that these are actually quite populous in the tongue. So we know that there is ACE2 in the tongue. So if the virus is coming in through your mouth, perhaps it is doing some damage in there. But we don't know that. As far as I know, there has been no work directly showing, there's been no RNA-seq or other work showing what happens if you take, for instance, an animal or a human and infect it with coronavirus. Does it alter these type of receptors? Does it change sort of the makeup of the tongue, those cells? And then what does that look like? So this is kind of the Wild West. Not really very much is known here, but just like some of the other thoughts, we could have direct damage happening for the nerve cells. You could have some inflammatory response that's happening, which could result in a loss of taste. There could be other cellular genetic changes that occur. And then we know that taste bud cells themselves do express this cytokine. So cytokines are kind of the start of inflammation. And sometimes you'll hear that cytokine storm. And that's sort of this whole range of changes that's going to occur in a cell to basically fight off any intruder. So we know that taste bud cells have that property. And that could be happening in response to the coronavirus infection. But like I said, we just don't really know that for sure. And this might be that it's not all the way across all of those different spaces in the mouth, for instance, but maybe just some. So if just some of them are altered, then you might have some tastes where you're unable to taste them and other things that are intact. And one other thing that's interesting, and I don't know too much about this, but we know that inflammation overall can reduce zinc, which is really important to the body. You need zinc to have sort of these cellular levels of communication stay intact. And we know that if you don't have enough zinc, your taste ability might go down. And so some people have done some early trials where if you give zinc, this does seem to help. And we know that this might inhibit the coronavirus itself by turning off some of its enzymes. And then if we can do this, especially in the mouth, for instance, like the very first site where the virus is infecting and replicating your cells, if you could give zinc and sort of turn that off right away, can that prevent the virus from spreading, for instance, like down the trachea into other sites, into your lungs? So there is some promising work being done there with administration of zinc. I don't know how big these studies are. I don't know if they're widespread, but they do appear to be positive. And then one other thing I just want to mention, because another thing that we keep hearing about in the news, of course, is all of these different variants of the coronavirus. And what does that mean? Are these going to cause more or less loss of smell or loss of taste? Probably not. I think from my basic understanding is that you would still get the same symptoms from infection, but the variants are going to cause more infections of more people overall. So you probably heard that as well in the news or in your own research, that these variants, they have different mutations that occur. So each one of these rays that are coming out from this kind of cartoon here, this is the RNA or the genetic material. Each of these rays are a mutation. And so some result in a missing amino acid where something gets deleted, and then some result in a change. And we see in this red area here, this is the part of the DNA or the RNA, excuse me, that results in the spike protein. But lots of mutations occur here and change amino acids. And so when you get changed amino acids, then you get a change in a protein. And usually that protein changes shape. So one of the things that's being studied quite widely now is, of course, what these variants do. And they seem to bind more tightly to that ACE2 receptor. So once the virus gets in, it's still going to do the same thing. The body's still going to respond in the same way. But it does seem like it maybe can get in easier with these mutations that are happening in these variants. So that's sort of a bummer in terms of nausea or dysgeusia. I don't think those types of symptoms will go away. But one of the good pieces of news that I learned is that the different vaccines do seem to be effective in kind of taking on these different variants, regardless of their mutations, at least so far. This is changing, of course, quite rapidly. And this little piece of RNA is kind of small compared to ours. I think it's about, I don't remember, and I didn't put my notes in front of me, like I said. But I do remember that the rate of mutation seems to be about 1 to 2 mutations or base pairs per month, which really is kind of rapid, but sometimes they don't do anything and sometimes they do. So we don't really know how to predict how many variants we'll see before we might be able to eliminate this virus. I did want to try to click on this link, which just shows something cool. There are so many different pieces of data now out there, but I thought this one was cool in terms of genetics. And so let's see if it'll work. This is an animation that's going to show us how these different variants are spreading globally across time. So the variants are like 19A, 19B, 20A, 20B, sort of these different colors. They start with this dark purple here first. And then the animation is showing us across, I guess it's kind of duplicated. The map here is, we've got 3 United States. But it's showing us the spread. And then you can see in each country or each region how those variants are changing or increasing with time. And so as the animation plays, the time is here. We started with 2019 December, and now we're going into 2020. And so this is China. And initially, we only had that dark purple, but now we have all of these different variants coming around. And you can see here in the United States. So these variants are spreading quite quickly and quite widely. Here's this latest one in red. And you can see that that is present as well in almost every region. So I thought this was a super cool website. It's very easy and sort of user-friendly, and you can do lots of kind of imaging or, data just mining with it. And it's called nextstrain.org. It also has some other types of viruses if you're interested in the spread of other viruses. So let's see if I can go back to my presentation here. And the last thing I think I just wanted to touch on was whether or not the vaccine would help prevent anosmia or dysgeusia or ageusia. Yes, the short answer is yes, of course. If you don't get infected, then you probably will not experience those symptoms, which is great. Some people have noted a temporary loss or temporary alteration in smell or taste as you respond to the vaccine itself. And so I'm sure you've heard this as well. Anecdotally, lots of people are reporting some symptoms right after they get the vaccine or maybe their second vaccine. In almost all instances, this is very short-lived and does seem to go away in like a day or two. If you have longer lasting anosmia, this could indicate an underlying infection itself. And so that's something that I think people might not realize, is that you can get the vaccine, but you may have already contracted COVID. And so you could have an underlying infection that results in anosmia. And it may be that it's not directly related to the vaccine, but you may just have it happening in the background. And with that, I think I will end. I have a list of references here, which I'll be glad to provide if anyone has questions. I tried to put full text links here to sort of each of the publications that I referenced. So that way you don't have to deal with any paywalls. And I really thank you for your attention tonight. And I'll take any questions.
Awesome. Thank you so much.
Yeah.
I think you can end your screen share and then people can just look at our wonderful faces. Wow, there was a lot of great information in that lecture. We have a few questions for you. One of them is wondering, is there a connection between parosmia and other type like synesthesia? Because you mentioned like within a smell getting crosswired, like can that happen where it crosswires between smell and other senses?
Yeah, I think so. And I don't know very much about like the mechanism behind that, but a lot of the deeper structures in the brain, like in that parietal lobe that we talked about, for instance, the amygdala is one brain region that's widely cited to have both olfaction and taste information. And so if there is any kind of dysregulation that happens there in the amygdala, that can result in a perception that's different. So I think that synesthesia could come about from some kind of crosswiring that happens in those deeper structures of the brain.
Yeah. Another question is about smell training. And I don't know if this is a nonsense question or not, but like if you're not anosmic, can smell training do anything for you? Like, can you learn more smells? Does it rewire your brain at all if you already can smell things?
I'm not sure. I mean, like I said, I'm not an expert in sort of these clinical aspects, but I would say it might help you in discrimination. Like if you already have sort of the hardware, you might be able to sort of fine tune it a little bit, right? Like with any practice, you can sort of enhance what you already have. And so I would imagine that smell training could help you do better with like discrimination, determining what things are different or stronger or weaker, but probably not like with detection. So if you couldn't smell lemon before, it might not add that for you. Again, I'm just speculating.
Oh yeah, I mean, this is great. We're here for you to speculate with us. There's another related question also about olfactory training. Is it possible someone could train their smelling incorrectly if the smell sticks are mislabeled?
Oh, I think
the worst thing to do on somebody.
I think maybe, but I mean, I do think there's so much other converging information. So I guess if you really couldn't detect like lemon again, for instance, at all, then it may be that you could sort of tell yourself that's orange. And then, you might forever think that that's orange. But I think for most of us, I guess if it wasn't labeled, you wouldn't know, right? Of course, but hopefully you have somebody who's sort of helping you with this, either a doctor or a practitioner, who would then say, well, think of lemons or think of some other context, like I mentioned, and then that would help you sort of prevent those errors from occurring.
Okay. And don't forget, folks watching at home, put your questions, if you have any questions, put them in the comment section, either of Facebook or YouTube, depending on what platform you're watching on. We're still taking more questions. we're just getting a lot of questions about olfactory training, so I hope you're feeling okay with this, but how do they choose the smells? Is it like based on the molecular structure? Is it common smells? Is it kind of random?
I don't know, to be honest. I really don't know, and I didn't really delve into that. I mean, I know when we study, odorants in the lab, a lot of times it is based on chemical structure, and you want sort of the simplest thing possible so that there's no chance for error, or you want things that are very different from each other. I would think for smell training, perhaps they want something that's generally known. So, it depends maybe too on your culture, like what you might be exposed to. And that might be an interesting, like UK. I mean, UK might not be that different from America in terms of cuisine, but I would think like cuisine might make a difference and some other things like that. So maybe those come into play.
Okay, we have a question about memory. In like thinking about how much our memory is connected to our senses, I guess I've heard about with Alzheimer's patients, like using music to help them get their memory back. Is there anything like that with smells to help maybe like regain or retain memories with Alzheimer's patients?
I didn't really come across that in this sort of report in my research for this. I didn't think about other senses sort of helping to jog your smell senses, but of course we know that smell is one of the most powerful, provocative memory stimulators. If you smell something, you're immediately transformed sometimes to another place or another person or some memory. So I'm not sure if you could kind of work that backwards, for instance, like see that person visually and then the smell sort of is evoked.
Yeah.
Seems plausible.
Yeah. And I guess, how do they measure in your, like with brain scans wise, that connection between smell and memory? Is it just if your neurons are active, or like, I guess, how do you track that?
Yeah, I think a lot of times, when we try to study humans, we don't really have as many tools as I alluded to. Like we can't really just cut open everyone all the time to check things out, which is a bummer. But so a lot of things have to happen in kind of an MRI situation. And so you can say, like, think of your grandma and smell this at the same time, or whatever, your two things are. And then they see which 2 or which 7 or which 27 brain regions would sort of become activated. And there's some pros and cons to that, because activation doesn't always mean that is the only thing that's happening. Of course, like your whole brain is working all the time. There's these complicated networks. And then, as I sort of wanted to talk about a little bit in this talk, they're very tiny little genetic changes that can have big impacts. And so those wouldn't be detectable in an MRI situation.
If someone loses their smell during a cold or COVID-19, has there been cell death? And when smell returns, have new cells been created?
Yeah, so that generally is the thought. And that is one thing that is super cool about smell in particular, is that those olfactory sensory neurons and a lot of the supporting neurons, they're turning over all the time. And researchers think that is due in part because it is sort of out there, like literally, this is out there right in the world, and it's susceptible to all this damage. And so it sort of has to be constantly able to regenerate, or else we would probably be anosmic very early on, just from onslaught of toxins and things happening. So if you do regain your sense of smell, it probably is because all those cells are kind of new and regenerating, and you hope that they're wiring themselves up correctly.
Let's see. Are people who work with zinc a lot, like galvanizing or doing moss treatment, that kind of thing, would they be less susceptible to coronavirus?
I'm not sure about that. And
it's just like around you rather than ingesting.
Strong suit. I want to say no, because I don't think that's how it works. I don't think it's that easy. The way that we have to ingest stuff to get it into our cells, and especially into our brain, is very specific. It's one of the reasons like vitamins don't always do what they're supposed to do. And so I don't know if like, just handling something would result in like infusion into your cells in a way that would be meaningful.
Do you know, and you mentioned this a little bit with the zinc and coronavirus. So if you were ingesting zinc regularly in a useful way, would that be like a reasonable preventative measure for coronavirus?
Yeah, I think so. I mean, I think there can be, some instances where you can like overdo it on things. Of course, you don't want to take too much zinc, but I think there's no downside to sort of taking it as a preventative, because that way you might help some of these deficits and then prevent some of those things from happening if they are zinc mediated.
Oh, this is great. I'm glad somebody else asked this question because I had it in the back of my mind too. There was this TikTok thing. It became very popular. I'm not on TikTok, but somehow it filtered its way into my old person media, I think through NPR, that charring an orange on the stove, you like char it so it's completely black and then you peel it and eat the orange will bring back your sense of smell and taste. Have you heard of this?
No, I didn't come across that in my research. I did not consult the TikTok, but I don't know how that would work unless like, maybe there's some change in the chemical composition, but it seems unlikely to me. I mean, I have kids, and so I have seen some things from TikTok become true that I was very like, that's not going to work. That's not going to happen. So, I mean, I guess, if you have an orange around and you're careful, like you could try it.
Yeah. Let's see. Oh, hold on. I've got to read through a few things here. Some people taste cilantro as dish soap, which I feel very sorry for those people because I love cilantro. It's the best. What's that related to? Is there a smell equivalent of that kind of weird thing?
I think that is sort of this taster, non-taster thing. So there is a specific gene, which some people have one or, allele or the other, or if you have both, maybe you're sort of in between. And then you can kind of hate cilantro or not. And I agree with you. It's very sad if you hate it.
Because does that kind of gene allele thing happen with smells at all?
I don't know. I should know that, but I don't know, to be honest. I didn't come across it and I don't remember it in any of my sort of years of having this all filtered through my brain, but it could be.
And maybe this is related, this next question, but there are some smells that some people find amazing while others find them awful. Is smell idiosyncratic?
Yeah, sure. It's very subjective. And some of it has to do, I think, with, context and our other senses and where you maybe first came across something. I think there are some things that we sort of agree across the board are noxious or dangerous, like the smell of gasoline, for instance. We all kind of cognitively know that that's a toxin, but lots of people enjoy it subjectively. So I think smell is very subjective.
Yeah. Okay. I have a weird personal story question for you that I hope is relevant. I used to live in Denver and I lived really close to a giant Purina puppy chow factory. And when I first moved there, it was terrible. And I couldn't handle the smell of the dog food all the time. And I didn't even notice when it stopped, but I think it wasn't too many months. All of a sudden I didn't notice it anymore until somebody visited me and they would mention it. Is that like related to cell death? Is that just not reaching, is that a receptor thing or a brain thing where that's
kind of this thing called desensitization, which you may have heard before, and we talk about it with lots of other things as well. But it is kind of a training thing where you still have those cells and those receptors, but your neurons have fired so many times in response to that puppy chow smell, but some part of your brain is like, I get it, stop, turn it off. And so it's kind of this dampening that happens over time when we're exposed to the same stimulus, right? And so you sort of become like saturated and your brain can't really handle that activation anymore. So it kind of turns that part off. If you were to go away for a while again, like this just happened to me, I went away for like 4 days and then I came home and I was like, my house really smells like, I forgot how my house smells. And so it can kind of come, it can be renewed. It's still there. Your brain's like, oh yeah, that smell. Ooh.
Cool. I was pretty thankful for that at the time that I didn't notice it anymore. Someone says here, they noticed that coffee tastes very different after eating a few orange peel pieces or peppers. Do you know the science behind this?
So that has to do, again, with kind of this desensitization that happens and some crosstalk that happens between taste receptors. And that's why, when you go wine tasting, for instance, they always offer you something in between to sort of turn off the activation that just occurred and then reset it. And so, yeah, there is some receptors which will respond to taste sense are a little bit more simple than odorants because there's not as many classes of receptors. So you can imagine something that's complex like wine or coffee is going to activate lots of different things in your mouth. And then you kind of have to turn those off or you'll get that same desensitization where your brain's like, I get it, coffee, wine. And so it's helpful to add something new, to sort of turn that off, reset it, and allow you to taste, something again.
Is that why coffee is often used as like a smell palette cleanser? I've seen it places because it is like just complex and different. That's kind of the criteria. Or
yeah, and it's very strong. I mean, it is one of the things that it has like a lot of really volatile odorants that come off it, and most people find it sort of pleasant, which I think is also important because you wouldn't want to offer people like a dirty diaper as your palate cleanser. I mean, definitely not your palate cleanser, but like even your odorant palate cleanser.
Yeah, I was just gonna giggle about that.
Right.
Okay. A smell doesn't return for maybe like years, some really long period of time after a cold. Would smell training still work in that case, or is it more for if it's been a shorter term loss?
It seems to me in what I read that it might not work in those cases because then you really might have permanent damage that's not just at the level of those olfactory or the epithelial cells that do regenerate. But if you have anosmia that lasts for a long time, it might be that the damage is like sort of deeper in these other neurons that don't regenerate. And so we know you always hear like if your brain cells die, they don't really come back. And that's true for almost all of the neurons, except for these very specialized ones that are sort of right at the forefront. So if those guys die back there, then you might not be able to do anything about it.
Okay. If COVID-19 virus attaches to the ACE2, could a person who's taking an ACE inhibitor for hypertension reduce their chances of getting COVID-19?
Yeah, that's a really good question. And that's something I had sort of, and I forgot to mention it, but we, the doctors of the world have seen that quite often, that does seem to offer some protection. So in fact, there have been some who suggest giving it sort of prophylactically and then some who don't. And there's a big disagreement there about whether that's, because it's very widely used, of course, for hypertension. That class of drugs has been around for a long time, has really proven, safety record, but we are always sort of cautious about giving things when we don't need them.
Yeah. Oh, that's really cool.
I know. I mean, I have high blood pressure and I take one of those. I was like, yes. But then I have high blood pressure. So I'm already like in this category of people who are sort of susceptible. And I'm not sure. I think that is a net wash.
Yeah. Okay, folks, we are winding up. If you have any last questions for Dr. Lockwood, please put them in the chat. Otherwise, I just want to say I'm going to pass on a few comments of thank you again so much for this talk. It was really informative and enjoyable. And I loved all the science visuals you showed. I get like really dorky into those, so that was nice.
Yeah. So yeah, thanks again to everyone for all your great questions. That's kind of fun and makes me think about, what I should have included, what I should learn more about for next time.
Always be improving.
Yeah. All right.
Okay, everyone, it looks like we are just about out of time. I hope you all enjoyed tonight's event. If you would like to watch this video again or share it with your friends, or if you know someone who doesn't currently have access to electricity or internet, this video will stay up forever and you can check it out on the video section of OMSI's Facebook page or on our YouTube channel. And please don't forget to follow us on Facebook, Instagram, and Twitter for updates on future events and inspiring content from OMSI. And I know I mentioned it at the beginning, but please consider supporting Science Pub and making a donation via the Facebook donate button, or you can visit omsi.edu/donate. Please join us we won't be having Science Pub next week because we'll have that after dark with winter beers, but in 2 weeks from now, on March 2nd, we'll have a lecture with Dr. Scott Burns as he takes us through the geology of the Oregon coast, which is very exciting. Once again, thank you to our partner, Celestream, for making tonight's event possible. And as always, you can get more information on all of the things on our website at omsi.edu. So thanks, everyone, and have a wonderful night.